So again, my apologies that there's no pizza. Also, my apologies, our email server is down, so that's why there's no reminder about today either. But thanks to those of you who did have it in your calendars and made the effort to come. So this is the last of our Lunch and Learn lecture series for this term. So there's no more until after Christmas, and we'll look into, usually we take the sort of next term off just because we're doing a lot of demos and teaching in the facility. But we'll definitely start something up by the summer if we don't do next term. Okay. So what I wanted to talk to you today for the last one of our talks that are kind of in our introductory session is just talking about live cell imaging. It's kind of interesting because this is something that we actually don't do a lot of at the HCBI. I think a lot of it has to do with the clientele we serve. There's a lot of people on this side of the river that do a lot of thick dead tissue imaging. And a lot of the live stuff that's being done around here I think is often done in your own labs. Whereas if you go over to the Nikon facility on the other side of the river, it's the exact opposite. It's like live cell imaging is their bread and butter. But we do a little bit of it, so there's just a few things that I want to go through just so that you have this in the back of your mind when you are doing live cell work. The things that you have to keep in the top of your head. So what are the really important factors for when we're doing live cell imaging? So the first big one is usually temperature. Obviously, if you're working with mammalian cells, things like to be at 37 degrees. If you're working at something, a model system, it's sometimes somewhere in between there, room temperature. Sometimes room temperature is fine. Sometimes room temperature is an advantage if you want to slow down development of something. But temperature control is usually quite important. And we do this through a number of different means. We can do it through temperature stages, inserts, we can put a big box around the entire microscope, we can use ejector feeders. So there's lots of different options here. Another big one is humidity. And this is one that's sometimes often forgotten. And a lot of the devices that are used over on this side have no ability to incorporate humidification into them. If you're doing a short experiment, this isn't going to matter too much, but a lot of these are done in special sample holders that have a very small volume in them. That volume evaporates pretty quickly. It changes the concentration of the salts that are inside. As well, sometimes people who are doing bacteria work have a little agar pad on top of their sample or below their sample. That can dry out pretty quickly. So humidification is actually really important. And so this is something else that you need to consider, especially if you're doing a longer term experiment, multiple hours, multiple days. Something else that can change is the pH value. So most of the cell culture media and buffers that we use are all carbonate based, which means unless you're maintaining a certain level of CO2 in the environment around that buffer, the pH actually starts to change pretty quickly. So this is something else that you need to keep an eye on. Does your buffer require CO2 supplementation? And finally, there's a few things that I put in the other category. So some of these are mechanical, like maintaining focus over a long period of time. And then some of them are also problematic when we're looking at fluorescence imaging. So things like photopunching and phototoxicity. So this is kind of the main sort of areas that I want to hit on today. And I just want to point out a few of the features of a standard live cell microscope. This I shouldn't say standard, this is kind of a pseudo. So this is really similar to the cell observer microscope that we have in the facility. So the first thing that you might notice is this big plastic box out here, and this big black pipe coming into the back of it. So this is a heater. So all this is doing is just blowing hot air into this big box. The larger of a volume around your microscope that you can get to the temperature you want, the less you're going to have to worry about thermal drift, expansion and contraction of the various materials within the microscope. Of course, the ultimate way to do this, and I've seen lots of labs that do this, is just turn up the temperature in the entire room to 37 degrees. Not the most environmentally friendly way to do it, You usually have to have a pair of shorts and a tank top at work, but the more of the microscope that you can encase in the temperature that you want to work at, the more stable your experiments are going to be. So that's the purpose for this big box around the outside. But along with that big box, we often also have a heated stage insert. So this is actually two pieces here. There's a black lid on the top, and then on the bottom there's another piece of black metal that's plugged into the stage. And that's an actual heated insert, so that whole insert heats up as well. So that just gives you a little finer control. I talked about humidity before. So what we have here is just a bottle of distilled water. And there's an air line that's coming out of the CO2 mixer here that I'll talk about in a second. It's coming in, it's just blowing bubbles in this water so that the air gets humidified. It comes back at the top through the line and into the back of the microscope over here. Sorry, actually right into the lid here. So into the back of the microscope and then right into the lid. And that air comes all the way out around your sample. And that just keeps your sample humidified and keeps it from drying out. Down here we have a CO2 mixer. You can also get oxygen mixers as well. And this is going to, there's another line coming from the CO2 tap in the facility into the back of this box. And it just mixes CO2 with whatever percentage you tell it to along the there. And then that's what goes in and bubbles through this humidification bubble, or bottle, and then into the microscope. And the last thing that is actually not on this microscope, but you'll see it on the one in the facility, there's a little box that sits on the side here. It's a little camera and an LED, and that's what Zeiss calls a definite focus module. This is a focus maintenance module. Other companies have them. Nikon uses perfect focus. So they're all slightly different in the way they work, but the idea there is just to maintain your focal plane within your sample while you're imaging over time. So if there is focal drift in the system, which there always will be, some expansion and contraction, those are going to be corrected for by the focus drive. So here are the big things that I want to hit on today. Just sample prep. What are the differences with sample prep when we're talking about live cell imaging? How to choose the right microscope for a live cell imaging experiment. What are the imaging parameters that you need to control and adjust to make your live cell experiment work the best it can? And then just real quickly at the end, I'll talk about a few of the specialized techniques that we're able to do with live cell samples that we can't do with big samples. So sample prep. So there's a whole number of mounting options. Usually the first one that everyone walks in with is some sort of tissue culture plate. There's a lot of problems with these. We can image these, but your image is never going to look very good. The biggest reason for that is the very bottom of these plates is made up of 1 to 2 millimeters of plastic. That exceeds the working distance of most of our high resolution objectives. And it's also, the light just doesn't pass through there very well. So it does some poor things with the phase of the light as it goes through the plastic. So techniques like EIC or phase contrast aren't going to work when the light doesn't pass through the plastic. Or not work as well. So we try to avoid those if we can. Some of the other options that we have out there are these little 35 millimeter round dishes that have a cover slip inserted in the bottom. So this is a little glass cover slip right there that sits in the bottom of the 35 millimeter dish. And you can see this is the exact same thing in the 6-well plate. So each one of these has a little cover slip that's been inserted into the bottom, and you can grow your sample directly on there. Another one that we often see are these chambered cover glass slides. So up at the top here, these are LabTech 2 is the name of these. And you can get these ones that are called chambered slides or chambered cover glass. This is a really important distinction. You want the chambered cover glass. So these are the ones that just have a cover slip on the bottom and the chamber sitting on top of them. Culture your cells directly in them and just image with an inverted microscope from the bottom. These chambered slides, there's a few problems with these. One is, this is an actual microscope slide on the bottom, so if you try to come with an objective from the bottom, you're going through a millimeter of glass. Again, it's too thick for most objectives. The other problem is if you were, say, doing a fixed cell experiment, you can fix your cells in here and you can actually pop these chambers off. You can just pull them off. But there's some glue. You can see these dark lines underneath all the chambers. That's kind of like a glue or sticky tape that holds it on. And when you put a cover slip on top of that, the distance between your cover slip and your cells that are actually sitting on the slide is again usually greater than the working distance of your high resolution objective. So oftentimes the objective ends up running into the cover slip before you actually get to a focal plane within your sample. So try to avoid these as much as you can. Yeah, and just make sure you get these covered glass. Another option that's out there, I don't think I've ever seen anyone use these in the facility. I've used them before though, and they're pretty great. I like them a lot. So you get sort of a metal ring that a cover slip can sit right into, and then you have this little threaded insert that goes in the top. There's usually a little rubber O-ring there as well. And this allows you to just throw a whole bunch of cover slips into 6-well plates or into even a big 10-centimeter dish, throw your sample on it, and then just pull them out one at a time and throw them into one of these setups. These would fit in our universal plate holders, so those are definitely options as well, although I've never seen anyone around here with them. And then one last thing that's often used as well for maintaining humidity inside your sample is something called pulp foil. So we have some of these little adapters upstairs in the facility, and again, it's just a little metal ring. And you take a piece of this membrane, it's called pulp foil, it's a commercial product. You put it over top and then put a sealing ring on top of that. And this pulp foil will allow gases to exchange through it, so CO2, oxygen, but it won't let water through it. So this keeps your sample nice and humid, it doesn't dry out, but you still have gas exchange across it. And so I showed you the multi-well plates up above that have the little cover slip in the bottom and the 6-well plates. What if you want more wells than that? Very good question. Obviously you can't put, nobody puts little cover slips inside 96-well plates or 385 or whatever the ones are. So there's a couple options here as well. You can now buy multi-well plates with a thin cover slip bottom on them. And there's a few different ways that comes. So you want to make sure that it's a nice thin bottom. Sometimes it's hard to find this number. Most 96-well plates that you buy won't say anything, which in general means it's 1 millimeter polystyrene. But if they are meant for imaging, they'll often say optical glass, and then they should give a measurement of how thick that glass is. So what you're looking for is something that either says number 1.5 thickness or 170 microns, or something close to there. I find that there's a lot of variation in the 96-well plates as far as this number here, but you want it to be as close to 170 as you can get. And they come in 3 different types. So they'll come just with a standard cover slip glass bottom, which is basically the plate is just stuck to a big massive cover slip. And those are pretty good, although a lot of people's samples won't grow on glass. You can get them treated with polyol lysine, polyol, all that sort of stuff, but often people will have trouble getting their samples to grow on there. Something newer that's come out now, it's actually much cheaper than the glass, which is kind of nice, is polymer film. So it's a thin plastic film that goes on the bottom. It's about 150 microns in thickness. And it actually ends up being quite flat and it seals quite well. And these are a really good option. So I know Corning sells them and a few other companies. You can also get a thin plastic bottom as well. So there's a company called Antibodies that probably a lot of you have heard of. They're always at every trade show. So they have a special polymer plastic that they use that's very good optical properties. And I know a lot of the groups who have had difficulties with certain types of stem cells that just won't grow on glass. They seem to grow really well on this Antibodies. The one drawback is they're pretty expensive. Cool. So something else you have to think about is the actual culture medium that you have on your sample as well. And obviously the best option is to use whatever you're using in the incubator, but sometimes that's not possible. So a couple things that run into trouble is the biggest thing is autofluorescence. So this is just a standard bottle of DMEM. You see it has this red color from the phenol red that's in there. Phenol red's just there as a pH indicator for you. It's not needed. And you can actually get this DMEM, you can order it without phenol red, so just a clearer solution. The phenol red has incredible autofluorescent properties in the green range, so if you're trying to look at GFP in cells in phenol red, it usually looks terrible. Just all kinds of background. So just swap that out for the phenol red free DMEM before you come into imaging. More than 94. Another thing that has autofluorescence is serum proteins. So the serum that you're mixing in with your DMEM is going to create a lot of autofluorescence as well. So if it's possible to reduce the amount of serum or even go serum free just before the experiment, that's going to give you a little better contrast ratio, a little better signal to noise as well. Then the other consideration is, do you need CO2? So is this a carbonate buffer system inside your medium? There are a lot of carbonate-free buffer system media available out there, and sometimes that's an easier option, and then you don't really have to worry about CO2. If you are using one with a carbonate buffer, then you definitely need to use a microscope that can supplement CO2 into your sample. Okay. So then, what are our options for staining? Obviously, trying to immunostain an intracellular epitope is no longer an option because they're not going to be able to get that antibody inside your cell or inside your tissue. So we have to look at some other staining options. The one that's most commonly used, obviously, is fluorescent protein tags. You can still use fluorescent antibodies if you have an extracellular epitope, or if you're looking for something in the vasculature or in the bloodstream. You can also fluorescently tag ligands, so if you have a specific ligand that binds a specific receptor, something like EGF, you can stick a fluorescent tag directly onto that. And you can also use certain cell permeable small molecule dyes, so some of the DNA stains that are able to pass right through the membrane. And others. So what are the best fluorescent proteins out there? We've had a lot of people that have been trying to push to 4-color fluorescent protein imaging in the facility recently. We've tested out a whole bunch of different proteins, and this seems to be sort of the best complement right now that we found that works with most of our microscopes and still gives decent signal in all 4 channels. So in the blue channel, this M-tag BFP2 is a great molecule. It's nice and bright. You can see it has a pretty good brightness rating. It's quite photostable as well. This can be excited with a 405 laser or a DAPI filter tube or a BFP filter tube. EGFP is still a great protein. It works well, fantastic in the green channel. TD tomato is kind of my favorite in the orange-red range. It's a lot brighter than M cherry, and it's more orange, which means you don't have to worry about sort of spectral separation in the red, far red range. And then more recently, there's this new molecule out now called IRFP, infrared fluorescent protein. And you can see it's not nearly as bright as the others. However, if you do compare this to some of the other, like, more traditional dyes that we've used in the past, especially the ones in the blue range, it's fairly comparable. And the other nice thing is that the infrared light doesn't scatter as much coming out of your tissue. So if you have something that's a pretty thick tissue, a few cell layers thick, maybe some organoid spheroids, you might even get a better image in this channel than this channel, just because there's going to be so much less scatter of that light coming back out of your tissue. Okay. Some of the other ones that we use that are still obviously very good are cerulean is another cyan color, citrine is a yellow, YFP derivative, M orange works quite well, M cherry is still pretty good, and MK is sort of, in some systems it's definitely, you can mix this with the TD tomato, so you can almost replace the far red with the MK if you're using a system with a spectral detector like one of our Confocals. Cool. And this is just something that I've shown before. So these are sort of our favorite dyes for 4-color experiments. So something in the blue range like DAPI, Alexa 405, or BB 421, Alexa Auto 488, Alexa 568, and Alexa 647. Cell tracker dyes, these are small molecules that can penetrate into your cell. Once they're inside the cell, there's an enzymatic reaction that does not allow them to come back out, so then you can wash all the excess dye on the inside out. So these are good fluid filling sort of cell volume markers. You can mix different cell populations using them. There's a number of live cell dyes for nuclei staining that will actually pass through the membrane and allow you to stain the nucleus. So one is hooks, which a lot of you are probably familiar with, very similar to DAPI. Another one that you might be less familiar with is CyberSafe Green. So this is marketed as a non-toxic nuclear dye. Take that with a grain of salt. It's still circulating in your DNA, so probably not super safe. But we have done some studies where we put this on developing mouse scales that were sort of in an in vitro culture or ex vivo culture. And the tails seem to develop quite well, even with this mixed with culture. So not much different from the tails without dye. And I think we've done that on a couple other model systems as well. Definitely less of an impact in the sequence works. And then finally, this is getting a little old, but these snap clip tags, there's now a whole bunch of different similar ideas out there, and this is basically you just genetically engineer a short peptide sequence on the end of your protein of interest. So instead of putting a bulky fluorescent protein, it's just a short couple-free peptide. And then you take a dye that's conjugated to one of these short chemical tags, put that on your cell, it can pass through the membrane inside, and then it actually finds that little peptide sequence on the end of your protein of interest and binds right to it. So what you get is a nice intracellular staining that you can do as well, but you get all of the advantages of using a dye instead of a fluorescent protein. Cool. The next thing is choosing a microscope. So the first question is always upright or inverted. Inverted microscopes in general were developed for live cell experiments. That's usually where they excel. But there are some situations where an upright microscope is going to be a little bit better. So one example is if your samples won't grow on glass, you really need to have them on plastic in sort of petri dish arrangement. You can leave them in that petri dish and just bring your objective in from the top. If you have a really thick sample, you can now start working with, you can't really see this very well, but some of these water dipping objectives that have nice long working distances. Something that you can't get a very long working distance, high resolution objective on an inverted microscope. So if your sample is very thick, this is often a good idea. If you have something where environmental control isn't as important, so say you're working with zebrafish embryos or cricket embryos that develop pretty well at room temperature, and you don't have to worry about CO2 and heating and things like that, an upright is a good option. You need to access your sample from above, and you can't use a cover slip. So those are reasons to use an upright microscope. Reasons for an inverted microscope are usually when you need really good environmental control. You have thinner samples that are growing really well on cover slips, or you're doing some sort of specialized technique like turf that requires an inverted microscope. Which objective are you going to choose? So we've talked a lot about objectives in this series in our Lunch and Learn. In fact, Sebastian did an entire seminar just on objectives. And the one thing that we always talked about is numerical aperture. We said, don't worry about magnification. For the most part, magnification doesn't matter. What really matters is the numerical aperture of your objective. And the higher your numerical aperture, the better your resolution, the more light you're going to gather, and the better the image you're going to have. This is the one situation where that's not true. Okay? So in live cell imaging, it becomes really important to match the refractive index all the way through your system as best you can. So you have cells that are sitting mainly in water, and there's a refractive index of about 1.33, and your cells are sitting about 1.36. So if you put an oil immersion objective under there, you're now going into a glass cover slip that's about 1.4 1.5, 1.52, and then into immersion oil that has the same refractive index as that. And what that means is there's a big refractive index mismatch there, and you've got to get a lot of spherical aberration. So if we take a look at the exact same sample with an oil immersion objective and a water immersion objective, you can see the numerical aperture is lower on the water immersion objective, so you'd expect a worse image. But it's actually a lot brighter and a lot higher resolution. And that's just because you have a better refractive index match throughout the sample. Okay? So this is the one situation where it becomes more important to match that refractive index than we're in good numerical aperture. A couple other things to consider if you're using some of these water dipping objectives like we have on the upright microscope. The size, the diameter of this objective is quite large. And if you're getting into the clear tissue ones, it's even bigger. If you drop this into a 35 millimeter dish or a 6-well plate, your sample has to be dead center because you can't move that plate anywhere without hitting the objective. Okay? So oftentimes with objectives like this, it's better to work with 6 centimeter or 10 centimeter plates. Then you have some more wiggle room in your sample. All right? Another option, if you are stuck with these tissue culture plates with the really thick plastic bottoms, we do have some objectives that can work over longer working distances. So this is a special, you can't see here, it says LD, it's a long distance objective, it's a 20x. This can actually image through that millimeter thick bottom and still quite deep into a sample. But you'll see you give up something for that. So the numerical aperture here is only 0.4, with the normal 20x, you're usually at 0.8 objective. The other thing, when you're trying to cover this many wells, you're not going to be able to use an immersion objective anymore. If you try to use an oil immersion objective, once you get about 5 wells down, all of your oil is smeared off across that plate, and you don't have any left. So this is a situation where you're going to be stuck with air objectives. All right. I said focus maintenance is really important as well. So this is an example of this in its worst situation. You can see these are images of the exact same area, just taken a few seconds apart. You can see that sample is slowly drifting out of focus. So this happens quite often in lifestyle imaging because we do have a lot of expansion and contraction due to thermal processes in the equipment that we're using. So it's very easy for stuff to drift out of focus. So on both of our 880 confocals and on ourselves or our system as well, we have what's called the definite focus unit from Zeiss. Essentially what this is a little box that's bolted onto the side of the microscope. It has a camera chip in it. And then it also has an LED. Okay? So this LED passes through a grid. That grid is projected up through your objective. And wherever there's a refractive index mismatch, that grid shows up. Okay? So this ends up showing up right on your sample in between the glass cover slip and the water. And that image comes back down and then goes into this camera chip that's sitting on an angle. So what that means is because this camera chip's on an angle, only one of these grid lines will actually be in focus. And then what the system does is just every couple time points or every few seconds, whatever you program it to do, it just double checks to make sure that that grid line is still in focus. This works okay. There's definitely some areas of it that could be improved. And there's been a new system that's just been released last week actually. So hopefully we'll have that one in the facility in the new year. And it'll be a little more reliable. But for the most part, this is a method for keeping your sample in focus. There's also some other methods that are built in the software too, just doing a simple software autofocus, so just doing a Z-sec, finding where the best contrast is. Those are options as well, and you can combine them as well.
So is this as good as, so if you notice there's a focus drifting, so and then you manually, if during your experiment you cannot do the autofocus, and then you just manually do it, is it still okay?
It's still okay. When this is working well, it's better. It's faster. It usually does it a little more often than you sitting there doing it yourself.
Yeah.
So when this is working well, it's better. It is very sample dependent. There's just some samples where, as far as I can tell, this grid just doesn't reflect back very well, and it will just fail on those samples. I've been told the newer version of it's better. We'll see. The one that I find works really well is Nikon's Perfect Focus. So it's a little bit different. It takes an infrared laser beam and just bounces that back off your sample, and it comes back onto a quadrant-based detector. And as soon as that reflected laser bounces off the middle of that quadrant detector, it readjusts for it. So it's definitely the gold standard in the industry right now. And that one's fantastic because it can actually run continually. So this one can run continually as well, but you sometimes get, especially if you get into a red far right channel, you'll get some bleed through this LED that you're using.
Do you know if the new system in the new year is going to incorporate that new technology or a modified version of this?
So they just released, so they're calling it Definite Focus 2.
Yeah.
They just announced it this week. We have, our cell observer is up to be replaced next year on our Evergreen agreement. So that would probably be by the summer that would be here, and it will have our 880 with Airy stand is also up to be replaced next year, but that probably won't be until like the end of the year by the time it gets here. Again, cool. So another choice that you have to make when choosing your microscope is what are you going to use for illumination? So there's a number of different ways for illumination. We can do just white light. So this is when we're doing Widefield, DIC, phase contrast. We just have a low power little white halogen bulb in the microscope, and we're just passing that through our sample. We can use wavelength specific LEDs. So we have a few systems that use these, like our cell observer. Obviously, we can use lasers to excite our sample, or we can use a white light lamp. So this is either a metal halide lamp, or now we're switching over to mostly like an LED array to get white light. And then along with the filter cube. And what you can see is that there is a greater potential for bleaching as you go across here. Take this with a grain of salt. You can get a really powerful LED that'll bleach more than a white light lamp if you're working in an area that white light lamp doesn't have a lot of power. But in general, as you move to the lasers and the white light lamps, you can get a little more bleaching in that direction. Another thing that's very different from microscope to microscope is the type of detection that's used. So all of our Widefield systems are using cameras, and the cameras come in different flavors. So you can just have a standard CCD camera. You can have a more sensitive EMCCD that might allow you to do lower exposure times, keep your cells a little happier. We have CMOS cameras that can do really fast exposure times that will allow you to get better time resolution in your sample and also shorter exposures. But we also have a number of point scanning confocals, and here we have all of your light concentrated into a single point and then scanned back and forth across your sample, which is slower, and then it goes back onto a detector, which might just be a standard PMT, or it might be a gas detector, which is a little more sensitive. It might be a non-V-scan detector if you're doing 2-photon imaging. And obviously, the more sensitive that detector is, the more potential you have to lower your laser power and not improve your sample as greatly. Cool. And then one last question when you're choosing your microscope is, are you going to use Confocal or are you going to use a Widefield microscope? When it comes to live cell imaging, the thing I try to default with everyone is to go straight to Widefield if I have a sample that I think has any chance of succeeding with Widefield. Widefield's a lot easier. You have the option of deconvolution to get a little sharper image, and it allows you to image much faster and be a lot more gentle on your cell when you're looking at live cell imaging. So unless you desperately need confocal, i.e. you don't have a really thick sample, let's stick to Widefield. So obviously if you have a thick sample, you're going to need confocal just to get that optical sectioning and get rid of that out of focus light. The drawbacks to confocal are they're usually a little bit slower, but I'm going to show you a couple slides on how we can get around that and speed them up. And then, of course, we now have this wonderful technology called light sheet, which is sort of this perfect compromise where you get almost the speed of a Widefield microscope, but you still get confocal sectioning comparable to a confocal. So, when it comes to actually setting up your experiment, with a fixed cell sample, it doesn't matter too much. The biggest thing is if you turn up your laser power too much, you're going to bleach it before you get it all imaged. But with a live cell experiment, you really have to think about this a little more intently. And there's two things that can happen, and these terms are usually used interchangeably, but they're two very different things. So photobleaching is when you hit your sample, you hit your fluorophore with some light, it goes into a dark state, eventually does some bleaching, it can't fluoresce anymore, right? So you can bleach your sample, but your cell might still be happy. Your cell's still happy, it's still growing, it's still dividing, but it's not giving it fluorescence anymore. On the other hand, phototoxicity occurs when the molecules, the biological molecules that are inherent to your sample absorb all of that excitation light that you're pouring in there, and that does some damage. So obviously if you're hitting your sample with near UV light, you're doing some DNA damage in there. And so in this situation, you can still have lots of fluorescence coming out of your sample. Your cell still looks nice and bright, gives you a really great image, but your cell is dying or near dead or already dead. And that's because of this phototoxicity. So these are two very different things. Often they occur sort of synergistically and at the same time, but there are lots of situations where you can have bleaching with no phototoxicity or phototoxicity with no bleaching. And so what you end up doing is a lot of compromises when you're setting up your imaging. And there's really three things that you have to compromise on to prevent bleaching and phototoxicity. So one is your temporal resolution. So how fast can you actually image? If you're imaging really quickly, that you're imaging a high number of frames, and that means more light exposure to your cell. If you space out your time points, you get lower temporal resolution, but now there's less light exposure to your sample, and your sample is going to be happier for a